So you can see in this image where you have the yeast that are budding apart of each other, and that makes these cells completely identical when you have this type of asexual reproduction. Here's a microscopic image of a yeast called Candida albicans. If you look here, this is that chlamydiospore I was telling you about. These little round structures that you see. You can also see the pseudohyphae, which are these extensions here that you're seeing off of the organism. And then you can also see the budding yeast cells, also known as blastospores, in these circular fashions. So when you look at this under a microscope, unless you have, good familiarity with what it is you're looking at, you kind of would be looking at it like, oh, well, this just looks like a big bunch of circles. But then over time, after looking at so many slides of these particular organisms, you would come to understand exactly and clearly what these structures look like. So with yeast also are pretty much found everywhere. They're a type of fungus. We also have yeast on our skin in various regions of our body. If you've heard the term yeast infection, a lot of women can end up acquiring yeast infections through improper hygiene in their the genitalia region. You can also find different yeast, though, in soil, in water, and pretty much, like I said, all over. So these have been very instrumental in our food industry, and they've also been instrumental in our antibiotic treatment industry. With yeast, the primary one that we work with is called Saccharomyces cerevisiae. Which is a yeast that we pretty much use all the time in baking. The Candida albicans is the yeast that's known for basically vaginal infections. So anytime you have, or anytime women experience, complications with urinating or burning sensations, a lot of times we refer to that as a yeast infection. Because your yeast is a part of your normal, the yeast is such as Candida is a part of your normal flora, and then sometimes that just gets out of place in certain parts of your body. And in doing so, that, can cause complications and irritations for the patient. Yeast colonies are easy to detect, I think, looking at them on a petri dish, just because of the way that they grow. But for somebody who's never worked with these cultures in a laboratory setting, sometimes it can be very hard to distinguish between the two different types of organisms, between the fungus and the bacteria. But the way that we do differentiate is by simply looking at the organisms underneath the microscope. The yeast, as I told you, it looks more like an oval shape, and it's a lot bigger than a bacterial cell. And a lot of times when you look at yeast underneath the microscope, you can also see that asexual reproductive process of budding. And bacteria, we know, do not bud. Instead, they undergo a process called binary fission, where they essentially split apart, which is also asexual reproduction, but it's not going to be to the scale of what we would see if you were looking at yeast under the microscope. Here's an image of the Candida albicans that's grown on blood agar. It looks like what you would see if you just had some bacterial colonies growing on it, but if you actually had this in front of you, you'd be able to tell that the fungus itself looks a little bit kind of more hairy-like and fuzzy-like than what bacteria actually look like underneath the microscope. Here's again another image where you have both bacteria, you have yeast, and then you have your blood cells all incorporated into this image. And what you can see here is what we want to do is identify where the bacteria are and identify where the yeast is and where the white blood cells. Now the white blood cells are going to be all of these cells that are stained with the light pink in the background. This is a Gram stain, so it uses safranin to stain the stain the specimens on here. It also uses crystal violet. So crystal violet looks like it's been uptaken by our fungal cells. Here's our yeast located here. And I know that because these are very oval shaped and a lot bigger in comparison to our bacterial cells, which are situated down here. And the bacterial cells that we see on here look like that they're going to be coccus in shape. But the bacterial cells are situated on here, the oval cells are situated up here, and everything else that's stained in red is going to be considered your blood cells that are inside of this sample here. So those are the three things that if you were working in a clinical laboratory setting and collecting samples, say from like a sample of urine or a sample of fecal material or just a regular blood sample, these are the types of shapes that you would look for to determine whether or not a patient is infected with a yeast or a bacteria, or to see how high the white blood cell count is in a patient, because that can determine how sick a patient is based on the number. Okay, so molds are also a group of fungus. There's two different spellings for it, MOULDS or MOLDS. It's not really very critical, but just so you know that this is not a typo, it is a MULDS. But molds can pretty much be found in water and soil. You can find them a lot of time in the fridge. You can find them pretty much anywhere where we don't employ very good housekeeping techniques, such as cleaning or cleaning with bleach. Most molds use filaments that are the same as all the other fungus we've talked about, except for yeast, which are the hyphae, and that allows the mold to have structure to it. Molds have both aerial hyphae and they have vegetative hyphae. Vegetative hyphae help with anchoring the organism to a substrate. So if you've seen mold growing on bread before, the mold is able to adhere to the bread because it uses the vegetative hyphae to anchor itself to the bread. Now, all of the fuzzy mold on top of the bread is going to be the aerial hyphae, where you have dark black spores that you typically would see situated on the very top of the aerial hyphae. And you can see these pretty distinctly if you've ever seen mold growing on bread or growing on pizza or growing on anything that was left in your refrigerator for a long period of time. Reproduction for molds is going to occur either through sexual reproduction or asexual reproduction, and with spore formation, this is going to take place on the aerial hyphae. So again, if you see mold growing on bread, it's probably the organism called Rhizopus and at the end of the aerial hyphae filaments, you will see those black spores ready to release spores into the air to give those fungal spores a chance to germinate on a new substrate or a new food source. Molds are very important to us because we use them pretty extensively in the development of antibiotics. Penicillin, if you ever have any fruit growing in or any fruit left in your fridge, such as oranges, you can see penicillin growing on there pretty distinctly. Penicillin has a green kind of color to its spores once it's germinated. So you'll be able to see that. There's also cephalosporin, which can be used as an antibiotic, and just a lot of different antibiotics that are, a lot of different fungal organisms that are used in order to produce antibiotics to treat infections in patients. We also use molds to produce different enzymes, so specialized enzymes that can be kind of taken in the form of a pill, can be given to somebody maybe to undergo or initiate the process of weight loss or help in digestive properties. So we can use molds in that capacity too, to use them as a way to produce large quantities of enzymes that we can sell for commercial value. Certain cheeses, their flavor is determined on the presence of mold. Roquefort cheese, Camembert Camembert and Limburger cheese, all of those, as well as blue cheese, get their flavor through the presence of molds that grow in them. Now, fleshy fungi are fungi you're probably the most familiar with. These are mushrooms, so there's a typical stalk, and then on the top of the stalk you have something that's called a basidocarp, or a carp, which is essentially the reproductive sac where all of the spores are situated. But anytime we talk about fleshy fungi, we're talking about something that looks like it has a lot of kind of meat to it. Not meat in the sense of high protein, but it's a very thick fungus that is growing. And that entire structure consists of the hyphae that in mass collective form is referred to as mycelium. And so we can find fleshy fungi pretty much growing all over in the soil, and pretty much wherever there is decaying matter, such as on a rotting log, and where there is kind of a nice moist environment to allow those spores to germinate. The fruiting body is the giant portion that sits on what's called the thallus, or the stalk of the fleshy fungus, and in that body, as I said, that's where the reproductive structures produce gametes, and those gametes get released into the air in the form of spores. And we can ingest fleshy fungi, and then there's some fleshy fungi out there that we shouldn't eat because they have toxic properties, but you just have to make sure you know how to identify those before you go eating anything that you find out in the wild. Fungi, again, used in a variety of ways. We either like to eat it, cook with it, or we like to use it to treat certain infections. So diseases that are in humans, diseases in animals, diseases in plants, we know that we can initiate treatments, especially if they're bacterial in nature, by exploiting some of the properties that fungi have. So this is from yeast, molds, other fleshy fungi. All of those have a very great medical significance to them. And if an organism acquires an infection or some sort of disease from a fungus, then we refer to that as a mycosis, because the disease is by nature originating from a fungal organism. And fungal infections can either be superficial, they can be cutaneous, they can be subcutaneous, or they can be systemic. So we can have infection of fungal from fungus in a variety of platforms. And kind of to delve into the different types of infections that you can have, you can see superficial mycoses are infections that occur in the outermost areas of the human body. So individuals that suffer from fungal infections where you have fungus growing on hair, or fungus growing on nails, or fungus growing on the epidermis, those are going to be considered superficial mycosis infections. For cutaneous mycoses, what you'll see are fungal infections that exist on the living layer of the skin, known as the dermis. So this is going to be just below the epidermis, and that's why it's referred to as cutaneous mycosis. An example of this would be that of ringworm, where you have dermatophytes, which can basically get underneath your skin and that living layer, getting in the dermis tissue and causing ringworm to develop. Ringworm is interesting just the way that it gets its name, because it looks like a worm has basically dug in this layer of your skin and just sat and formed a ring. But it doesn't have anything to do with worms, so this is all a fungal infection that you could get. Yeast infections can also cause cutaneous or oral or vaginal infections. They also are examples of cutaneous mycoses. And one key example is that of the Candida albicans. For subcutaneous and systemic mycosis, these are very severe types of fungal infections. These can take place in the dermis as well as in deeper tissues underlying the dermis. One example is that of Madura foot. Systemic mycosins are fungal infections of the internal organs of the body. So we even have deeper infections with systemic mycosis. And anytime you have spores being inhaled, they can cause a lot of complications.